Water, it is everywhere, shaping life as we know it. But have you ever wondered how water flows into thirsty plants, or why soaking raisins in water makes them grow like magic? The answer lies in a fascinating biological process called osmosis. The secret behind how cells stay hydrated, plants stay upright, and even why pickles are so delicious. Today, we are exploring osmosis. So, grab a glass of water, and let's flow into the science of osmosis. Osmosis is the movement of water molecules through a selectively permeable membrane from a region of higher water potential or low solute concentration, to a region of lower water potential or high solute concentration. This process is crucial for maintaining cellular functions and occurs without the need for external energy. Osmosis is a type of passive transport, meaning it doesn't require any extra energy to occur. This happens because water molecules move along the concentration gradient, from a region where water is more abundant to a region where it's less abundant. Gradient is a measure of how much something changes as you move from one region to another. For example, from one side of the plasma membrane to the other. Therefore, a concentration gradient is defined as a measure of how the concentration of a solution changes from one place to another. Think of it like a ball rolling down a slope. The slope represents the concentration gradient, and the ball naturally moves downward without anyone pushing it. Similarly, in osmosis, water moves naturally along the gradient, from high water potential to low, balancing the concentrations on either side of the membrane. Unlike processes that require energy to move substances against the concentration gradient, like rolling a ball uphill, osmosis relies entirely on this natural downhill flow, making it energy efficient and essential for life. This process occurs naturally in biological systems and plays a crucial role in various physiological processes. Osmosis is driven by the difference in solute concentrations on either side of a semi-permeable membrane. The membrane allows the passage of water molecules, while restricting the movement of larger solute particles. The movement of water molecules continues until equilibrium is reached, or until the pressure difference counteracts the osmotic flow. The movement of water molecules in osmosis does not stop when the concentration on both sides of the membrane becomes equal. Instead, it continues, but at an equal rate in both directions. This state is called dynamic equilibrium. When equilibrium is reached, the water potential on both sides of the selectively permeable membrane is the same. At this point, water molecules still move across the membrane, but there is no net movement, because the flow in one direction is balanced by the flow in the opposite direction. In simple terms, osmosis doesn't stop, it just stabilizes. When discussing osmosis, it's important to understand three types of solutions. 1. Hypertonic solution. A solution with a lower solute concentration than the cell's internal environment. 2. Isotonic solution. A solution with the same solute concentration as the cell's internal environment. 3. Hypertonic solution. A solution with a higher solute concentration than the cell's internal environment. If an animal cell is placed in a hypertonic solution, it will gain water, swell, and eventually burst. The cell is said to be lysed. If the cell is placed into a hypertonic solution, it will lose water and shrivel up. If a plant cell is placed in a hypertonic solution, it will gain water. However, the cell wall is rigid, and so pressure builds up inside the cell. This prevents an excessive amount of water from entering the cell, and also stops the cell from bursting. When a plant cell is filled with water, it is said to be turgid. The pressure it applies on the cell wall is called turgor pressure. In annuals and young plants, the central vacuole acts as a fluid skeleton. If it is turgid or tight with water, the leaves are crisp and upright. If they lose too much water, they become floppy. If you soak up a limp lettuce leaf in water, the vacuoles fill again, and the leaf becomes crisp again. If a plant cell is placed in a hypertonic solution, the plant cell loses water and its turgor pressure, making the plant cell flaccid. When this happens, the plant wilts, but can recover if water is made available. Further, water loss causes plasmolysis, in which the pressure decreases to the point where the cell membrane peels away from the cell wall, leaving gaps between the cell wall and the membrane. Small single-celled animals, like paramecium, that live in fresh water have a constant problem of water flooding into their cells. They overcome this osmoregulation problem by having a small pump called the contractile vacuole, which gathers up the excess water and pumps it out of the cell at regular intervals. Fresh water. Fish are often in danger of drowning as water pours into their bodies by osmosis. They overcome this problem by giving out copious urine. Marine bony fish have a tendency to lose water because their blood is less salty than the sea. To manage this, the fish drink a lot of seawater and excrete salt from special glands in the gills. Seabirds and turtles also drink seawater and weep salty tears from special glands near the eye. Cartilaginous fish like sharks manage to keep the osmotic pressure of their blood slightly higher than the seawater by retaining some urea in their blood. In this way, they do not have to worry about water moving in or out. Plants absorb water from the soil through their roots, via osmosis. The root hairs act as a semi-permeable membrane, allowing water to move from the soil with high water potential to the roots that have low water potential. Salting or sugaring food draws water out of microbial cells via osmosis, preventing spoilage. In dialysis, osmosis plays a crucial role in removing excess water from the blood. The blood is separated from a dialysis fluid, dialysate, by a selectively permeable membrane. The dialysate is designed to have a lower concentration of water compared to the blood, creating a concentration gradient. Water from the blood moves across the membrane into the dialysate by osmosis, helping to reduce fluid buildup in the body. This principle ensures that excess water is efficiently removed, while the process of diffusion eliminates waste products and toxins. Together, osmosis and diffusion restore balance in patients with impaired kidney function. Now let us look at the major factors that affect the rate of osmosis. 1. Concentration gradient. The larger the difference in solute concentration between the two sides of the membrane, the faster the rate of osmosis. A steeper gradient creates a stronger pull for water movement. 2. Temperature. Higher temperatures increase the kinetic energy of water molecules, making them move faster, and thus increasing the rate of osmosis. 3. The surface area of the membrane. A larger membrane surface area allows more water molecules to pass through at a given time, speeding up osmosis. 4. Permeability of the membrane. The presence and abundance of specialized water channels, or the membrane's inherent permeability, influences how easily water molecules can pass through. 5. Pressure. Applied pressure, such as turgor pressure in plants, or hydrostatic pressure in certain systems, can either enhance or oppose osmosis, depending on the direction of the water movement. Thanks for watching this video on osmosis. Stay tuned for our next video, in which we will delve into other forms of cellular transport.